Good morning, everybody. Here is Copenhagen calling. I'm your chairman here in the first session, and we should perhaps better be going. Christian, you don't have anything to add? Okay, he is shaking his head. So the first speaker is Liang. We'll talk on re-exploring the biological affinity of chitinozoans, evidence from morphological variation and exceptional specimens. Go ahead, Jan. You have to unmute your microphone.
Okay, so can you hear me now?
We can hear you now. Go.
Okay. First, I want to make a test. I don't know how to share it. Okay,
So Jan, just click on the green share screen button, and then yeah.
Yeah. So now can you see my slides?
Perfect.
Yeah. Oh, okay. Then I will begin. Okay. Thank you, everyone. And you guys get up quite early. And so today I'm going to talk about the biological affinity of chitinozoans, and based on our recent study on morphological variation and exceptional specimens. And in this talk, first I will make a brief review of previous hypotheses related to chitinozoans, and then an introduction to our recent work. So as one group of the microfossils, chitinozoans are characterized by its bottle-shaped outline, and are widely used in biostratigraphy. However, their biological affinity is still debated. So generally, there are two camps related to the affinity can be recognized. One is linking chitinozoans to different groups of protists in the 1930s to 80s, mainly based on morphological comparison. And the other is the nowadays prevailing egg hypothesis. And around the 1980s, there also are some other hypotheses have been put forward, but with less acceptance. So since 1980s, based on the data collected and analyzed, some conclusive works are carried out. And for example, we thought that the sealed test of chitinozoans, which seems impossible for any communication between the inner side and the outer environment. And the lack of juvenile and reproductive specimens in the record, and there is no perfect analog in current biology. And with those discovery of those cocoon-like clusters, and more people believe that chitinozoans are eggs or egg capsules of an unknown group of meteoroids. However, in a recent study, three-dimensionally preserved chitinozoan species, the Hyracochaetina violata, shows a very significant morphological variation. It is clear that the test outline changes from the short and stout form into a long and cylinder form. And more interested, the distinctive ornamentations, which can be stably observed in both of those short forms and cylinder forms, which changes from simple spines in the oral part and to multi-rooted spines in the middle and the lower part, and to those jungle-like spines in the base. And a statistic study based on those specimens shows that all those specimens belong to a single group, but with a quite large size variation, especially in the test length. And the longest test is more than 3 times than the shortest test. However, the size variation in nowadays even eggs are generally within 2 times, which is significantly smaller than those observed in violata. So in order to test whether such a large variation is common in all chitinozoans, we collected the previously published measurement data of 365 species. And the result also shows that once enough specimens have been measured, the ratio of longest to shortest test is around 2.5, and 1/4 of them are more than 3. And the size variation of the eggs of extant or aquatic metallic species shows that the CV ranges from 0.7 to 16. However, the CV of chitinozoans of the Violala is as high as 21, and which is significantly larger than those than the size variations of those undisputable eggs. So it seems rational to assume that chitinozoans are not eggs, according to this work. And furthermore, in the same strata yielded the Violala, we got 2 of those exceptionally preserved specimens. They are characterized by less complete test attached at the base of a complete test. And in previous publications, those of exceptional specimens have also been reported in Ordovician and Silurian stratas. And Kramer and his colleague also even suggested that those specimens are record the gravity moments of chitinozoans and put forward a mode to show that, okay, how chitinozoans reproduce themselves. However, this work with less acceptance, and no further studies have been carried out, maybe due to the limited techniques in that age. So then those exceptional specimens have been taken as abnormal or teratological specimens for a long time. However, compared to those real teratological specimens, which are characterized by an irregular bulge or extension, those exceptional specimens are featured by their regularities. First is the directionality, that is, all the short and less complete tests appeared only at the base of the complete one. And then is the repeatability, that is the morphology of the short test is highly resembling the morphology of the complete test. And under the near-infrared microscopy, we can see clearly a shadow resemble the neck margin of the upper test, which appeared in the chamber of the lower past. And this kind of materials are quite fragile that is broken into 2 pieces when we carefully moved it for further test. The morphological features suggest that this specimen belongs to Benoni Katina Weissenbergensis with a test length of 86, which is the smallest size ever reported, and resembling the missing juvenile specimens in the record. And a few more exceptional specimens have been examined under the micro-CT, and those specimens are recovered from the middle and upper Ordovician of Baltica and North America. And the result shows that both the vertical and transversal sections shows that inside the chamber of the complete test, the neck of the upper test occurred. And we also got some another type of exceptionally preserved specimens, which have also been reported before in 1960s. And this type of specimens is featured by carrying a chain consisting of several small individuals, which become progressively shorter distantly. And under the micro-CT, we can see clearly, it is clearly that an opening can be observed at the center of the base of the last individual. And more interest, and it seems the open base could be closed at some time. For example, in the figure F, we can see the base of the lowermost test is already closed. And the micro-CT result F2, figure F2, we can see the lowermost, the base of the lowermost test is going to close, but not totally closed yet. And the other, the lower specimen presented in figure G. From this specimen, we can see clearly that the neck of the second individual also appeared in the chamber of the first individual, and the last two individuals' leg of a neck only in the chamber exists, as highly suggests that those individuals represent a different stage in maturity. Here we made a video based on the micro-CT data of the last specimen. Then we can have a better review to see a better view to see how those individuals are connected with each other, even the inner side. Based on the materials we examined and analyzed, two reproductive modes of chitinozoans are proposed. Is that one parental individual produces one or multiple offsprings at a time, or almost at a time. So for the first mode, that one individual produces one offspring, and this mode quite resemble budding, which is a quite common sexual reproduction strategy in some earlier branch animals and plants. And it differs from budding in its directed orientations, that the clone only take place at the base of the parental individual, and the other mode, which produces multiple offsprings in a chain, resembles strobilation. And this strobilation is a real asexual reproductive process occurred in some certain cnidarians. And one of the main differences is that once the chitinozoan offsprings detached from the chain from the parental individuals, it was already a mature independent individual. But the strabila produced by the strobilation need further development to become an independent organism. And at the same time, ultrastructure and morphological studies on the reproductive specimens revealed some quite interesting details for the first time. For example, the plaques in the juvenile test, which is in figure F and G, and the plaque is quite thin, less than 1 micron, such as in figure J. And it seems to be composed of many rounded granules. However, the normal plaques in chitinozoans, because plaques are quite distinctive and important morphological structures for chitinozoans, generally it should be either quite thick or very complicated.
2 minutes.
Sorry. And also we can see that the hollow in figure N, because I don't have the mouse, so I can't point. I can show you that in figure N and figure O, we can see a hollowed copula. And this is quite distinctive because this kind of structure have never been observed or reported in the genus Triatochatina. And also we can see the carina that is marked by the red arrows in figure O. We can see the carina is not empty. Inside it is filled with some fibrous structures, and this structure is also have been, it's the first time to show it quite clearly. And also one more quite interesting thing is the sponge-like materials, which marked by the white arrows in figure D. And also it can be appeared in the inside of the chamber, such as in the yellow arrow in figure Q. And this kind of sponge materials are quite common on those reproductive specimens, and they can be appeared as fluffy sponge materials, such as presented in figure L and M, and all like dendritic or nested structures, such as in figure R and D. And also we can clearly see that those structures can be uniformed more and more closely and tightly, and finally become to form the condensed test wall. And you can see it in figure D. And the ultrastructure shows that both the fluffy sponge materials and the condensed test wall are formed by those clustered rounded particles, which are shown in the right two figures. And with those particles are with a diameter around 10 to 100 nanometers, which should be the basic materials forming the Katian test. Okay, so then here comes the main points and result. So according to this study, we found that those generally thought the abnormal specimens turns out to be the key specimens recording the reproductive moments of chitinozoans. And those rounded particles revealed for the first time, they are in nanometers, and they are the basic materials construct the Katian test. And the reproductive modes of chitinozoans resemble budding or stabilization, but with its own distinctive features, and generally belong to colon. And at last, according to the geometric outlines of chitinozoans, the significant variation within the species and the widespread distribution and the newly recovered reproductive modes, it seems more reasonable to link chitinozoans to an independent group of protists, but not X of anything else. Okay, so this is my talk. Thank you.
Thank you. Sorry, I was not clear on how much time we should have for discussion. We now have 3 minutes. Any questions? Everything was clear. You had a lot of data. No questions? We probably better have to stick to the time frame. So yeah, Joe?
Question. Yes, I'm just wondering how much variation there is in the normal size of a species of protist. You compared with eggs, and assuming they're one's biological species, that's good. But what about protists? Do they vary much more? Does an individual species of protist vary in size to the same degree that you see here?
Not actually. We didn't analyze the size variation of the protist because nowadays I also want to check, but this kind of study, the size variation of the eggs of any protist, this kind of study is quite rare because in nowadays people, the current biologists focus on those protists. They are more care about how those protists can do something good to us, or how they make diseases for fishes. But the pure study focus on how those protists reproduce, or how those larvae looks like. This kind of studies not too much. So the size variation of our study, we only compared it with nowadays invertebrate, such as the echinoderms and some others invertebrate groups, including 45 species, and not involved those protists.
Okay, thank you.
Thank you.
Thank you. Well, I can introduce the next speaker, then we are probably on time. It is Juwan Jeon, I hope I got that right, who will talk on Two Silurian-type stromatoporoid genera from the Upper Ordovician Beiguoshan formation of North China and their tectonic and faunal implications. I think you can go ahead, Juwan.
Thank you. Hi, this is Juwan Jeon from South Korea. And before I start, I would like to make I should make an apology because now I'm currently in a hotel and for the quarantines, and everybody are using internet. So somehow here the internet connection can be poor, so please understand it. And I am a master's degree student in Nanjing Institute of Geology and Paleontology. It is my great pleasure to meet many superstars today. And I will gonna present about Two Silurian-type stromatoporoid from Upper Ordovician section of North China. I would like to start with brief explanation about stromatoporoid or Ordovician stromatoporoid. During the Ordovician, there were two major different orders. First one is order Lavicida, which is the first stromatoporoid group emerged in the early Florian. Their diversity peaked in the late Ordovician. The second group is Claustrodictid. This group firstly showed their appearance in the Middle Katian. In case of Claustrodictid, its diversity peaked in Silurian. And present study is about Katian, the Middle Katian, which both stromatoporoids occur. In terms of their internal structure, those stromatoporoid groups have significantly different structures. First, Lavicid stromatoporoid is composed of pila and cyst plate, while Claustrodictid has laterally long lamina and short pillars. Katian is a very important stage for Ordovician stromatoporoid. As we can notice in this diagram, they reach the highest diversity peak and other new groups emerged during the Katian. However, somehow North China was insufficient late Ordovician stromatoporoid records, and this is why we start this research. Our study area is near Xi'an, an old city. The representing Upper Ordovician strata in this area is Beiguoshan Formation, which occurs widely in North China. The study area is Tiwadian section located in northwestern of Xi'an city, and the age of the formation is Middle Katian. The outcrop contains many limestone quarries, but now it is all closed. The Beiguoshan Formation contains pebble to border limestone conglomerate. They are interpreted as gravity floor deposit, and there are many stromatoporoids, tablet coral, rugose coral, and other sessile organisms. In the outcrop, we can easily recognize various organisms such as chitinozoan sponge and tablet coral and also rugose corals. Stromatoporoids are very common, especially those well-laminated Claustrodictid stromatoporoid, and they have been known as the major reef component. Until now, only 4 genera have been known from North China during the Katian. We additionally found 6 different genera, making a total 10 genera in North China. And those 2 red-colored genera are what I want to emphasize today. We can group those stromatoporoid into order level as Lavicid and non-Lavicid stromatoporoid. So let's see the first group. The first group is Lavicid stromatoporoid. They are characterized by the Declivolithus North China provincial Lavicid assemblage. These species have been spread to other neighboring regions of South China, Australia, and even Siberia during the middle to late Ordovician interval. The second group represented by sharing very, very few affinity with other Perigondwana areas. In addition, Flexodictyon and Simplexinodictyon have not been reported from Ordovician until now. More detail, Simplexinodictyon is characterized by its zigzag-shaped lamina which interfill the paralamina. The picture in the right below shows a typical species of Simplexinodictyon from the Silurian of Gotland. Simplex Synodictyon show big and stout internal structure, and it is easily recognized at outcrop. Their internal structure are distinguishable by its widely spaced lamina and short post-like pillars. This genus has been thought to derive from clathrodictid stromatoporoid. Previously, those two genera have been known to make their first appearance in Zuzlok and Wenlock of Silurian. Our finding extends their stratigraphic range to the earlier times of the Ordovician and the existence of their incomplete fossil record. Here, these fossil gaps are interpreted due to Huayuan hypergeny and inaccurate systematic works. In addition, those two genera are thought to be typical Silurian types and have an implication on fauna transition. First, Huayuan hypergeny can be divided into two events. In terms of our study, it is related with event 2, which make a big unconformity from late Ordovician to late Mississippian in North China. It is considered due to regional sea level fall and tectonic uplift. Thus, we do believe that Huayuan movement destroyed the stromatoporoid fossil record in North China, and therefore a geographic gap occurs in the record of the two stromatoporoid genera. In addition, taxonomic works on stromatoporoid have been inaccurate previously. It is very common that people assign stromatoporoid with lamina as clathrodictyon or dorsal clathrodictyon without further taxonomic considerations. So during the middle to late Ordovician, North China has been thought to move outward from Gondwana. In the Revelian, highly diverse and endemic stromatoporoids occur North China, and they spread during the middle to late Ordovician intervals. By contrast, as other non-lavicid, the clathrodictid made their first appearance during the Katian of Australia and spread to other neighboring regions of Gondwana and even to Siberia and Laurentia Baltica. In terms of North China, it exhibits different paleobiogeographic affinity with other peri-Gondwana area in terms of non-lavicid groups. Those two Silurian-type groups only occurred in North China, and we interpreted that this pattern reflects the outward migration of North China during the late Ordovician. So these are the group of stromatoporoid which have been known during the Ordovician. And on the right part, the numbers are indicating stromatoporoid which occur each period. As you can see here in this slide, lavicates are most predominant group and highly diversified during the Katian. This was the highest diversity during the Ordovician to the end Devonian interval. By contrast, clathrodictid and actinostromatotid were highly diverse and abundant during the Silurian, although they make their first appearance during the late Ordovician. The last group, stromatoporoid, was diverse during the Devonian. The majority of clathrodictid become major reef component during the late Ordovician as what they did during the Silurian. Although other two groups have originated in the late Ordovician, they become far dominant during Silurian and Devonian respectively. It seems that stromatoporoid fauna transition already occurred in Katian stage, and the late Ordovician extinction accelerate the transition as larvae kits significantly decreased during the Henantian. So it is the conclusion part, and here we report two Silurian-type stromatoporoid from Upper Ordovician Beiguoshan Formation of North China, together with North China Darwinian provincial larvae kit assemblage. This unique species assemblage possibly reflects the outward migration of North China from Peri-Gondwana area during the late Ordovician, and their incomplete stratigraphic gap from the uppermost Ordovician to the middle Silurian is interpreted due to the regional level 4 and the tectonic uplift of North China, which is well known as Huaiwen hypergeny. The finding of these genera is very significant in terms of stromatoporoid because that the Silodevonian stromatoporoid stock have their root in Ordovician. And for the last slide, I would like to express many thanks to Dr. Ursula Thum and Dr. Olga Orba because they provide me many valuable reference from Estonia and Russia. And I know that it is really time-consuming work, so I would like to express many thanks. And in addition, they also provide me an opportunity to check stromatoporoid type specimens from Estonia and Altai. Thank you very much. And for audience, thank you for your attention. So this is end. Please feel free to ask any question and critical comment. Thank you.
Thank you. We have plenty of time for questions. Any questions? Joe? Joe, we can't hear you. You have to unmute yourself.
I wasn't actually asking you.
Ah, you just waved a hand there. I can see another hand. Alejandra?
No, that was just a poll.
Thank you.
Lucy?
So
So Arne, we have a comment in the chat from Heidi Allen. She asks Juwan whether he can let us know about the dates of the Beiguoshan in the Lower Ordovician.
Actually, the Beiguoshan formation age is widely accepted as Middle Katian because of the conodont. But unfortunately, in our section, the study section, I couldn't find any conodont from here. So we try to make many thin sections, as many as I can. So we already make more than 1,000 of thin sections. It includes corals and stromatoporoid sphincterozoan sponge, and also for sedimentary structures. And in case of stromatoporoid, unfortunately, it cannot indicate specific age. So we believe that the sphincterozoan sponge, with also same species of sphincterozoan sponge occurred in Middle Katian of New South Wales. And in case of corals, we couldn't find any Silurian corals, and they are quite typical Ordovician North China fauna. So I do believe that it is Middle Katian without any problems.
Okay, thanks.
Any other questions?
Thank you.
6 minutes. Helen?
It's actually Ian. My wife's got the registration for Zoom. Hi, Arne.
The surname was familiar to me, yeah?
Yes, but it's just I'm masquerading as someone else. Juwan, I realised that Barry Webby found a Plexidictian, which you've got queried in the charts that you had. Would you like to comment on what that particular Plexidictian might really be, or is it just an enigma? I think it's only 1 or 2 specimens that were identified as that from New South Wales. It's an interesting connection with North China if it's an Ordovician record of Plexidictian. Thank you.
Thank you. So as already Dr. Ian Penshable mentioned that Professor Webby already reported one of the species of Plexidictian before, but at that time I think that he was almost impossible to check the specimen by his hand because some demoplastic problem. So I think that he just described as doubtful Claustrodictian. So yeah.
Any other questions or comments? We still have 4 minutes. Everything was clear from your talk.
Thank you very much.
Christian, don't you think we have to stick to the time frame?
Well, maybe Lucas will go overtime. We don't know, so we could start with it now.
Okay. Then the next speaker will be Lukáš Laibl, who will talk on a new Lower Ordovician nektaspid euarthropod from Morocco. Go ahead.
Thank you. Can you hear me? This is actually the first time I'm using Zoom. So
we can hear you and we can see you.
I'm actually happy that we are starting a bit earlier because I'm borrowing the Wi-Fi from my neighbor. So I hope he's not going to find out until the end of the session, at least. So okay, I'll just try to share my screen. Okay. Can you see that?
It's perfect.
Fine.
Perfect. So I'm going to show you some nice fossils, some nice arthropods from Morocco. This is actually our collaborative work with people from Lausanne. I've been working with them for more than 2 years. Alison Daly acquired quite a big collection from Fezuata. So we have plenty of fossils currently in Lausanne. And so this is one of our first paper based on this, based on stuff from this collection. So it might sound a bit strange. I mean, I don't know if you are familiar with nektaspids, but if I show you this picture, you realize that you probably are, because they contain some iconic Cambrian animals, such as Naroya from Bruges Shale or from Xinjiang, but they also contain some other less known forms, such as emucarids from Emu Bale or from China. And then a group called Leavids, which is actually the group that I'm going to talk about most. And then they also probably contain this strange trilobite-like creature from Sirius Passage from Greenland that was originally described by Graham Budd. So in the terms of their phylogeny, how nektaspids are related to other arthropods. This is one example from John Patterson, published in 2010. And you can see that nektaspids are actually closely related to some other Paleozoic forms, such as Helmetia or Tegopelti or trilobites, Zonderellids, etc. So they are generally closely related to trilobites and similar arthropods. And currently, well, the phylogeny is still not clear. There are some other phylogenies published quite recently that shows a bit different topology, but in general, we can say they contain 3 subgroups, 3 families. They contain emucarids, which are known from China and from Australia, then naroids, which are known basically worldwide, and then leavids, which are known mostly from Perigondwana or from Baltica. So, as I told you before, I'm gonna be focusing this talk on Moroccan leavids. They look like that. So these are some examples of leavids that has been described before. This is the first one is genus Pterycoia from Ordovician of Sardinia. Then we have Livia from Cambrian of Poland. This is Buenaspis, which has been included into Leavidae by some authors, but it's not quite sure that this form is a leavid. Buenaspis is from Cambrian of Greenland. And then the last one is Sumaspis from Sum Shale from South Africa, also from the Ordovician. So probably the most known, at least in terms of number of specimens and preservation, is genus Pterycoia. So this is species Pterycoia arusensis that has been described by Wolfgang Hamann and some of his colleagues in 1990, and it has been described from Upper Ordovician strata of Sardinia. You can see it has rounded or elliptical head shield or cephalon. Then it has, and this is quite interesting, it has 4 thoracic segments, although most of the times you can see 2 or 3. But if you look at this, the specimen that is in the right side, you can see that there are 2 segments quite clearly visible, and then 2 more segments that are hidden under the cephalon. And this is quite interesting. And as you will see, this is something we can see also in our Moroccan material. And then it has like a rounded or sub-pentagonal pygidium with a very pronounced keel or carina in the middle. So this is Sardinian stuff. This is just a view from Sardinia. And now what is quite contrasting is this a view on a Fezoata. So our new leavid comes from Fezoata. You are probably all well aware about Fezoata. This is just an overview. So Fezoata is located in Moroccan Anti-Atlas. This is the main area where most of the localities are located. The material we have described comes from approximately, can you see the cursor? Yeah, okay. So it comes from this part that is quite close to a village or tiny city called Benizoli, and not far from city of Zagora. And stratigraphically, it comes from the lower interval with exceptional preservation from the Arne group to Murai zone. So this is how the material looks like. It's quite rare. In our collection, there are 2 or maybe 3 specimens. There are some more specimens in the collection in Yale. this specimen has been actually, this species or this form has been actually reported in 2013, I believe, by Peter van Rooij in a GSA meeting. But after discussion with him, we now think there might be two species. So this is one species, and the second one is the one that was reported by him. Because the second one is stratigraphically a bit younger and does not have the pronounced carina or the keel on pygidium. So we named this species Taricoya because it resembles the Sardinian Taricoya arusensis, but because there are some clear differences, such as the shape of the cephalon, a different shape of the pygidium. I will show you at the end the comparison, so you will see the differences quite clearly. So because of the differences, we decided that we will name a new species that we called Taricoya tazagurtensis. It's because it's from Fezuata, and Fezuata is close to Zagora, and it was discovered by Berber family, and in Berber Zagora is called Tazagurt, so that's why we call it Taricoya tazagurtensis. So this is the new species of Taricoya, and new species of Lavid from Morocco. If we look closely at the specimens, this is the specimens we selected as a holotype. You can see quite clearly the cephalon here. There is some kind of ridge in the cephalic margin, in the anterior cephalic margin. Then you can see quite clearly the pygidium, which has the anterior margin pointed forward and rounded posterior margin, which is this is probably the main difference when you compare it with the Sardinian species. It has quite clear median keel. And in this specimen, you can see at least three segments. Then what you can see is this raised strange structure under the cephalon, and I'm going to talk about this structure a bit more in the next slides. So this is just a regular photography. This is immersed in ethanol, and this is of course interpretive driving. This is the second specimen. Again, you can see cephalon with a kind of like pointed or angular genal angles. You can see the pygidium with very nicely preserved keel. And then you can see 2 segments here. And then we think this was very difficult to decide. But now I'm quite sure that there are 2 more segments under the cephalon. And it's quite comparable with the Sardinian species. So we actually think this species also have 4 thoracic segments. And again, you can see some wrinkled structures under the cephalon. What is also interesting is that please note that this species is quite small. So this is 1 millimeter. So the whole animal, even if you compare it with the previous, with the holotype, it's about 8 millimeters long only. So let's have a quick look on these raised structures under the cephalon. So if we look at it, we can see raised central area with some finger-like projections going on both sides. And so this is actually quite similar to some digestive structures that we can see in some other nektaspids, such as in Naroya. In the second specimen, we can see something a bit different, but if you look closely, now I see the photo is a bit blurred, but you can still see there are like these more or less crescentic raised structures. And so when you compare it with some other nektaspids, for example, with Naroya, we can see something similar. There are some finger-like structures going here, and then you have these like bent raised structures. And of course, this is a part of digestive system. So we actually think that in livids, in Tarichoiatus agoutensis at least, the situation regarding its internal anatomy may be quite similar. So we think that of course we are not sure about the detailed morphology, but we think that Tarichoia may be quite similar anatomically to Naroya. So this is just a reconstruction showing the diversity of the morphology of the livid morphologies. We have these two species of Tarichoia, the new one from Morocco, Tarichoiatus agoutensis. Then we have Tarichoia arusensis from Sardinia, Cambrian Libya, Ordovician sumaspis, and perhaps also Cambrian buenaspis. This is the age of the individual forms. And what is interesting that originally the Cambrian and the Lower Ordovician stuff are marine, while the Upper Ordovician are inhabiting some strange environments such as lagunal or brackish environments. So the last thing I'm going to talk about is a bit about the paleogeography of these forms of the nektaspids in general. So when you look at the Cambrian, you can see that most of the nektaspids are actually distributed along the equator, while this is contrasting with the Ordovician, where most of the nektaspids, in this case represented mostly by livids, are restricted to quite high latitudes of southern hemisphere. So we actually think that at least livids, even if you look at the Cambrian livids, they are, with the exception of buenaspis, they are known from quite high latitudes of in Baltica. So we actually think that livids preferred, or might have preferred, like colder seas. And moreover, some of the more derived members apparently migrated or preferred some non-marine areas, while originally this group was probably marine, because the Cambrian representatives and also the Lower Ordovician representatives are known from open marine communities. So yeah, that's it from my side. So I hope you like this new Cambrian arthropod, and thank you for attention.
Thank you. We have plenty of time for questions and comments. Christian, can you shift the speaker view so we can see everybody?
I think Lukas needs to stop sharing the screen.
Oh, okay, sorry.
Any questions, comments? We have 7 minutes.
Got one to start it off then. Normally with the Fezzuata, you find species are restricted to a few sites, at least the unusual species. Yeah. Is that the same case here, that they're both from one location, or?
Yes, actually, well, I mean, these two specimens comes from two sites, but they are located like several tens of meters next to each other. So like, yes, they generally it comes from one site. And that, I think that might be also the case, as you said, that the species are located, these rare species are located to one particular site. it's probably true for this for the species as well, because we haven't found it anywhere else. I mean, we spent some time. We went like last November with Peter van Rooij. We went to the new part of the newly acquired material from Mohammed Ben Mula. So we checked like another 4 tons in his courtyard, and we haven't found any other specimen. I know from Peter that there should be maybe 5 additional specimens in Yale collection. But because of the current restrictions with the traveling, we haven't been able to check them. I only have some pictures from Peter. But yeah, we actually think that there might be either there is some variation in the shape and the morphology of the Pygidium, or there are two species of this specimen. But yes, in general, you are right. They comes from more or less one site.
You think that applies to the Yale specimens as well?
So at least based on the information I have from Peter, they should be more, I think they are above Araneograptus murray. So that's the other clue. We think it might be additional species.
Yeah.
But yeah, without seeing the material, it's difficult to say.
Okay, thank you.
No worries.
Thank you. Any other questions or comments?
Well, Lukáš, I can just ask a question here. It was a nice talk. That's not a question, that's a statement. But the question, the paleogeography, it seems like in the Cambrian, they were distributed mostly in the lower latitudes, and then in the Ordovician, they ended up in the higher latitudes, if I'm correct. And so you say it's an adaptation to colder waters, but will you say that these groups then originate in the lower latitudes?
Yeah, well, do you mean now? Do you mean the group like the whole nektaspids or only the libids? Yeah, I would say they originated in low latitudes probably, but of course there might be some taxonomic bias as well, because most of the Cambrian Lagerstätten are from low latitudes. That's the point, because like Burgess shales, Sirius Passet, Imubale, Zhenjiang, most of the Chinese Lagerstätte are from low latitudes. So maybe we only know the low latitudes stuff, but I think there are some Lagerstätte that are probably not entirely low latitudes, but yeah, well, it's a bit difficult to answer. But yeah, why not? They might originated in low latitudes, because that's actually the place from which we have most specimens and most species from Cambrian.
A final very short question. No. In that case, we might proceed. The next speaker is Sofia Pereira, who is going to talk on the trilobite assemblage of the, I have to pronounce that, Declivolithus Fauna, the Katian of Morocco, a review of new data. And you have to tell me how to pronounce that genus name there. Go ahead.
Hi, everyone. Thank you for being there attending this meeting. You can now close the Instagram and Gmail tab. Don't give up on me. Yes, I know 2020 has been a tough period, but look at the Ordovician. It was a much tighter one. Everything happened during the Ordovician. We have a biodiversification event, and we have a truly big extinction, not like that little cleansing of the earth in the Mesozoic. And we have temperatures for all tastes, a global warming event, and a glaciation to finish in a big cold way. And finally, in the Ordovician, we also have many changes. A new ocean in here, a subduction in there, an amalgamation elsewhere, a paleobrexit, and a bunch of perigondwana terrains. Sometimes I think, how cool it would be if magically we had access to original Ordovician maps. How close are we to what it really was? Each study, each work is one missing piece, and this work is one piece more. So this history started on 1906 with a very peculiar trilobite. Jelisko, a Czech geologist and paleontologist, was the one who erected the new species, Trinucleus alfredi, based on a label left by the famous and quite unlucky Otomar Novak. He died prematurely, and thus much of his work wasn't published, being scattered among manuscripts and old labels. Many of us are doing the same, gathering data that we can barely publish, and one day will be gone together with our eternally ongoing work. Guilty. But as I was saying, Novak left a label with his name, and these fossils came from a structurally complex area, the Rochemittal block. The fossils were not very well preserved, and we'll come back to the Rochemittal block later in this presentation. Some decades later, Klupac described a similar form, but from the Prague Basin in the Baudelaire Formation, much better preserved, not being flattened like the Rochemittal specimens. And he erected a new species, Tetraspis novaki. This species and the previous one were considered to be synonyms by Pribilla and Vaniek, who erected the new genus Declivolithus in the 60s. This attractive trilobite was then included in this important monograph on Trinucleina by Hughes et al. And of course, they commented on its weird appearance. They call it a bizarre trinucleid with much longer genal prolongations, almost harpid-like. And the few authors dealing with the genus, they also commented on the distinctiveness of this creature, spectacular and unusually large trinucleid. Outside the Czech Republic, it was only identified in Morocco, and that's to there that we're going now. When we think about the Ordovician of Morocco, and particularly its trilobite assemblages, we invariably think about Jacques Detombe, who everyone knows. This French geologist started working for the Geological Survey of Morocco in 1950, and he is one of the main responsibles for the geological mapping of the Anti-Atlas and its lithostratigraphic scheme, and for collecting an incredible amount of fossil data, more than 2,000 fossil localities and thousands of fossils collected from all the groups, which is sent to many specialists 40 years ago, and part of that material is still under study, being missing pieces to better understand the faunal links between Morocco and other regions. Detombe got particularly interested in trilobites, and he did make some important works on them. Thus, the first mention of Declivolithus in Morocco was made by Detombe in 1971. Curiously, it was not in the Anti-Atlas, but in a small inlier, right in the southern border of the High Atlas, in the Skora region, which anyway shows more analogies with the Anti-Atlas than with the northern domains. This information was also republished with more details in 2006, but these works have gone unnoticed. Only in the last decade, the genus was finally found in the Anti-Atlas, coming from the famous El Qaid Rami area as a result of the intense commercial extraction of fossils. If we search on Google El Qaid Rami fossils, there are something like more than 300,000 results, especially echinoderms and trilobites. See the Declivolithus in here. When it first appeared, the dealers called it Nankinolitas. Later, they improved the classification to Declivolithus alfredi, the Czech species. And it started to be very famous in commercial websites. Everyone was aware of its existence in Morocco, but it remained unpublished, like many other occurrences from there, until finally Forti and Edgecombe got ahead in 2017. They considered it is a new species of this genus, and they described an assemblage of 4 trilobites, which they correlate with the Baudelaire Formation from the Czech Republic, and thus the Upper Baronian. Nevertheless, they located these levels in the middle part of Lower Tower Formation, which does not fit their age proposal. As no data is too much data, and we had these more diverse assemblage coming from the same levels, here we are revising it. This is our fossil site, Boflos, located west of Erfurt, right in the limit of the Anti-Atlas and most of the Declivolithus. Occurrences across the internet and commercial stores came from this band here. This site itself is basically a hole in the middle of nowhere. It is exploited by Lazarus Mu and his sons, and 40 and Dutchkamp's locality is told to be further east, but maybe it is the same with a different transcription of the father's name because of the reluctance of the Moroccan collectors to reveal where are they actively mining fossils. What once again difficults the geological and lithostratigraphic settings of some Moroccan published material. The stratigraphic placement of Boflos locality remains problematic because of its isolated character, a single trench in the core of a small anticline delimited by faults that exposes a lenticular intercalation of fossiliferous gray micaceous mudstones and sandstones, corresponding to a local biophaceous development which was recognized not only in this area, but also there in the scoring layer of the high atlas, just where the tombs found the Declivolithus. It is hard to properly correlate these local sequences with the anti-atlas formations, and the few other groups who presented, like these visceral graptolites, are not helping. So we do need to turn our attention to the trilobite assemblage. Juan Carlos started working in Morocco in the 90s due to an official Spanish-Moroccan collaboration, and since then he kept going and studying the Ordovician of the anti-atlas. The first time he noticed these levels was in 2010 when these trinucleids started appearing in fossil shops. It was already this same family exploiting this site. They started by the sandstones because they were the first levels in the anticline. And in 2015, Juan Carlos made his first campaign to collect material. They were about to change from the sandstones to a fossiliferous mudstones intercalation. This is how the fossils looks like in the sandstones, preserving their original convexity. It's impressive. And later, 2 years later, he made a second campaign collecting the same species, but mostly preserved in mudstone and thus flattened. Our work provided, in addition to the 4 species already identified by Forty and Edgecombe, 7 species more of 4 different orders, increasing the diversity of the assemblage to 11 species. This includes new occurrences from Morocco, the improvement of systematic knowledge of some taxa, new paleobiogeographic and biostratigraphic data, and a new species. In what concerns the previously identified taxa, we also have a slightly different opinion at species level, thanks to the very good preservation of specimens preserved in sandstones. The main problem of trinucleids is the amount of variability you have within a population. And if you only have 2-3 specimens, you'll never know how the 4th would look like. A typological approach is unworkable for this group. We need to adopt a more population-based approach. Maybe the Moroccan and Czech Declivolithus corresponds to the same species. We're still in it, and I don't want to bother anyone with systematic details, that's my job. But it's important to state how a different preservation can enhance different characters and thus potentially create what I call taphonomic species. Another formation or another locality, another species. But many times it is related to a taphonomic imprint in the specimens. And what is great about this locality is that having an intercalation of sandstones and mudstones, we have the opportunity to compare the same species preserved either in flattened or preserving their convexity. One of the main differences that justify the erection of Declivolithus titan was the greater organization of the pits in the Czech species, which have, for example, these inner rows organized in a sulcus. In fact, in Moroccan flattened specimens, we can see it due to the collapse of the genal lobe over the flank, but in convex specimens, we were able to verify that these internal pits have the same configuration of Declivolithus from the Czech Republic. Let's see another example, this time an elanid. Elanids from Morocco are like that big elephant in the living room. We know they are there, we're just pretending we're not seeing them, and fortunately we get these cool guys preserved in mudstones, so I was able to recognize it. I was like, oh, I know you, you're a smooth and informative creature. I have it in Portugal. It is a new species, and I hate new species. They're so useless until they finally appear in somewhere else. That's the case. And this is how it looks when preserved in sandstones. And it was essential to reveal new insights of a Gondwana lineage that were not possible for me to understand in flattened specimens. A last example of how tricky is the formation we'll never say this is the same species. That's why I'm so skeptical about proportions-based species when dealing with flattened material. Width-length ratios, arguments like more rounded, more vaulted, slightly wider, it is not that easy. When studying Moroccan material, I always remember this sentence of Shaw in 2000. The paradox of an uncertainly based species name attached to an extraordinarily well-preserved characterization collection. Most of the identified species are previously known from the Czech Republic, and the new data will allow to improve the systematic knowledge of some of them, but most important, they support that the strong faunal link between Morocco and the Czech Republic is still existing during the late Ordovician, and it probably is stronger than the faunal link with the Ibero-Armorican domain at this time. We do agree with the previous assignment of Declivolithus Fauna of Morocco to the Upper Buronian, Katian and Slice 2, made by De Tombe and Forty and Edgecamp, because in fact a significant number of species are known from the Baudelaire Formation. Anyway, we cannot exclude it correlates with Middle Buronian because there is also a significant number of species in common with the Zahorani Formation of the Czech Republic. Besides, although the most significant tags are more characteristic of the Baudelaire Formation, they were also described from the underlying Zahorani Formation, and their presence can be more related to a facies control than to the age. And besides part 2, the Iberian links, they are restricted to the Middle Buronian up to the moment. Anyway, the data supporting an Upper Buronian assignment is stronger, and possibly these levels correlate with the uppermost lower tower or even with the Upper Silurian Formation. To turn things more difficult, we cannot forget the Rochemetal block, where the Declivolithus alfredi occurs and was discovered the first time. We're not sure about the age of those occurrences in Czech Republic because in this graben, the Ordovician is almost entirely represented by a monotonous shale sequence, the Voltus Formation. The type locality of the Declivolithus alfredi is a richly fossiliferous black shell in the middle part of this formation. Its fossil assemblage is not too diverse and the material is poorly preserved, but 4 out of 5 species of this assemblage are present in the Declivolithus Fauna of Morocco. I wrote another number for mistake in the abstract. And just like in Morocco, the correlation of this unit with the lithostratigraphic scheme of the Prague Basin is problematic, with fossil data supporting that this fossiliferous unit within the Voltus Formation correlates not with the Baudelaire Formation, but with the Zahorani Formation. In any case, we're certainly in Katian times, and it is clear that during the Upper Ordovician, at least up to the mid-Katian, the fossil assemblages of the Czech Republic, Bohemia, Perunica, are greatly correlated at species level with those from Morocco. If Perunica is going somewhere, then Morocco will be chasing it. Their faunal links are still very strong during early mid-Katian times. In recent years, there have been several published data suggesting that we need to change something in these projections. Even if we plot Perunica in the Gondwana margin during the late Ordovician, it is still hard to explain the data because Morocco is there. Or maybe this is okay and we just need to rethink the role of the facies control in all these terrains. And that's it. Thank you and wake up.
Thank you. Very nice talk. We have plenty of time for questions or comments. Everything seems to have been crystal clear. Lukáš?
Sorry, I don't know how to raise the emoticon hand. So very nice talk. I completely agree with the strong similarity between Morocco and Czech Republic. And I just have one question because I visited that locality last November, and I found quite a lot of tiny cyclopidgids. So I'm just wondering, have you found something similar, like baby cyclopidgids?
I think you're talking about Cyclopidge rediviva.
Yeah, probably.
Yeah, it's done one. I don't see any significant difference to the one from the Czech Republic, described by Marek.
Me neither, but have you found some tiny ones? Because
tiny
I found
ones.
like some meraspids, tiny meraspid
Meraspids,
stages.
I don't think so, but I need to check again. I have some of the groups with me. Cyclopedes I don't have with me. They are with Juan Carlos. So I have the photographs, and I didn't check the scale many times. I'm looking to the morphology, but I will check. But I don't think they are meraspids in our collection.
Okay, perfect. If you want, I can send you some photographs too. I have the photographs, but I don't have the material. It's stuck in Morocco.
Okay. Okay, send me. Just send me. No data is too much data.
Yeah. Okay, thanks.
Thank you.
Any other questions? Just speak up. I can only see a selection of you. There are 3 screens with participants. Nope. We are 5 minutes early, but at normal conferences, people are always going over time. Maybe this is the way to do it. Everybody keeps their time. But in that case, we should perhaps proceed to the next speaker, which is Andrej Ernst, who will talk on early Katian bryozoan faunas of Baltoscandia. Andrej, go ahead.
I'll start the screen. Do you see it?
Yes, we see it. But you should make it as a slideshow, so it is becoming even bigger.
Now is it okay? Well, if you can hear me and see, I just start. In our talk, we speak about our preliminary results on study on Ordovician bryozoans, upper Ordovician bryozoans, mainly from the interval of late Zambian and early Katian. And the aims of our research is that mainly a taxonomic study of bryozoans from this interval throughout the Baltoscandia, an overview of local and regional bryozoan diversity from this interval, and their paleoecology and paleogeography. Well, our material comes from several localities in Estonia, Norway, and Sweden. In Estonia, we collected from classical localities from Vasalemma quarry, Ristna Cliff, Postaspea. In Norway is this Steinberg locality in Mjosa Lake, and in Sweden we collected material from the Kulsbeck quarry. Here you see the definition of the localities. Here are the Estonian localities: Vasalemma, Ristna, Postaspea, Kulsbeck in Sweden, and 2 Norwegian localities, Mjosa and Steinberg. We study Paleozoic bryozoans, and for their study we need oriented thin sections. It's a very complex preparation, and you see usually we need 3 orientations for thin sections: tangential section, longitudinal section, and a transverse section of the branch. And you can see how it does look on the tapeworm bryozoan: longitudinal section of the branch, transversal section of the branch, and the tangential section showing the apertures. Well, but for the study of the solid limestones, we cannot extract each colony of the bryozoan and make separate thin sections of it. So we use just large thin sections made parallel to bedding and transfers to it. You can see how does it look. The width of these samples are about 4 centimeters, and you see bryozoans cut in mainly longitudinal tangential section, and here you can see bryozoans cut in mainly in transversal orientation. The first locality is Vasalemma quarry in Estonia. Here are the limestones of the upper Zambian to lower Katian age. And Here are some sections showing taperstone and cryptostone bryozoans from the Vasalema formation, which is uppermost. Then in age is already good preservation of fossils and bryozoans, especially. And in the slightly older Sauer member and from the upper Sandbin, we see that the bryozoans are locally very abundant and diverse. This slide is almost completely full of various bryozoans, taperstones, branched taperstones, encrusting taperstones, cryptostome and cystoporoid species. And what we can find here, there are large colonies. They are interesting in their composition. You see that the size is extremely large for bryozoans, more than 10 centimeters. But what is interesting that such samples are composed of various species. Well, in the core of such sample, we found a cystoporoid species with many layers of encrusting colony. But outside, there are two different species which belong to taperstone and Estenioporine genera. So it's actually not a single colony, but a bryoid. Another location is on the Ristner Cliff. How you can see, this locality is composed of thin-bedded limestones, and these limestones are full of fossils. What we can see first, microscopically, there are large colonies of taperstone and Estenioporine bryozoans, Diplotypa, Dinolitis, Mesotypa, many of them. And if you look on the closer distance, we see some other kinds of bryozoans, branched taperstone bryozoans mainly, encrusting taperstones here as well, and many others. There's a thin section made in the transverse orientation to the bedding. You see here a lot of bryozoans, but also other animal remnants like gastropods, mollusk, brachiopods. But bryozoans are extremely rich and diverse here. The third Estonian locality is Porsaspea. It's nearby to Ristner Cliff locality, and looks similar to that with thin-bedded limestones, which contains also rich bryozoan fauna, some large species like this Halapora, but also such places full of smaller species. On this slide, you can see more than 20 colonies belonging to different species of bryozoans. And if we look at the thin sections, so we see great diversity and abundance, and also the excellent preservation of bryozoans. And what is interesting for this locality is that the diversity is different from sample to sample. So the overall diversity is much higher than the local diversity of each sample. In Norway, we have studied two localities. The first of all is Steinbeck, this road cut exposing this Steinviker formation, which is composed of very solid limestones, dark in color, but by microscopic visual inspection, we can discover already large bryozoans like this Diplotypa you can see here. And if we make a thin section, so we can have much more bryozoans. Here you see cryptostone bryozoans, encrusting taperstones, branched taperstones, encrusting taperstones here, and another sample with hemispheric taperstone bryozoans with again cryptostones and some other species. So this locality was never studied for bryozoans. We are just working on it. But another locality, Mjosa, we studied this locality several years ago and described quite nice fauna. This locality is Katian in age. And so this is how does it look with bryozoans, many taperstone bryozoans. But so the fauna is not completely described. We still work on it. And somewhere in the future we can show some results on it. The only Swedish locality I used it in this work is a Kulsby quarry. It's very famous for fossils, but completely unknown for bryozoans. This locality exposes Kulbys formation, which is upper Fenben to lower Katian age. And if you look on the samples, we see large bryozoans, different growth forms, mainly benched, but also some massive plants, and also smaller species. Here, fenestrate, some strange taperstorms. But the preservation of this fauna is not so good as the Estonian and Norwegian localities. The only bryozoan species which was described from this level in Sweden is this species. It was described not from the Kulsby quarry, but from the Antian quarry, which is nearby. And this species is found abundantly also in the Kulsby quarry. But how does it look in the scene section? You see large bryozoans here, and the matrix, which is full of smaller bryozoans, which makes a lot of diversity of bryozoans. And here we come to some problems when we want to count the bryozoan diversity. First of all, the great variation in their size. If you look on the sample left, you see they are of approximately the same size, these two samples, but here are 5 colonies in species. And on this image on the right, you see also more than a dozen various species, which are much smaller than these bryozoans. So such samples we can estimate in the field, but such samples we have to deal in the lab because we need a microscope. So there are two different scales how we can count bryozoans, and it makes some difficulties. Another difficulty is that bryozoans can be easily fragmented. As you see on the scene sections, these two fragments may belong to the same colony, and these two fragments may belong to the, also to the same colony. So it's difficult to judge whether these fragments belong to the same colony or not. Well, at the end, I would like to mention some possibilities to use bryozoans for paleogeography. This is an approach which is largely underestimated. Here's a curious species which occurs really worldwide. You can find it everywhere in the Baltoscandia. It's a cyclostome bryozoans called Quasar borealis. It's a very small species. This stem is in diameter about 0.5 millimeter, but in scene sections you can find it almost everywhere. In Europe and also in Laurentian, North America, it was found in the limestones of the Zenben and Katian age. So bryozoans can be potentially used for paleogeography, but this is largely unused. Well, at the end, I would like to summarize some results of our study that we have done in the last 2 years. The fauna of bryozoan fauna of Baltoscandia is very diverse and rich, and it inhabits various environments. Bryozoans show high variation in their growth habits and sizes, but even size variation and fragmentation of bryozoan colonies hamper the estimation of the diversity, make it difficult. And in many cases, we can use bryozoans for ecology, for ecological and paleogeographical studies. And this approach is largely underestimated. And thank you very much for your attention.
Thank you. Can you switch off your shared screen?
Yes, just a moment.
Any?
Well,
you still have a shared screen here.
Yeah, no. Yes. Yeah, I hope.
There we go. Any questions or comments? Caroline?
Hello. Thanks, Andrej. They're beautiful bryozoans.
Hello.
The bryoliths, is there any evidence of them rolling? I know some of the ones in North America show bryozoans growing on all the sizes, all sides, or are they just going upwards?
Yeah, some of them look rounded, so they have this shapes, but some of them are slightly domed. So I think maybe both. They can be rolled. Yeah.
Thank you.
We have plenty of time. More questions or comments? Remember, I can only see a little selection of you, so speak up. Everything seems to be...
I'll have another one.
Yeah, Caroline?
With the Cucurcella, which you said it's got it's worldwide. I mean, I seem to see it in all different sections. Why do you think this one is so widespread compared to other smaller smaller cryptostones and other ones like that?
Well, this species is really easily recognized, and since sections is very special, and bryozoans are difficult for taxonomy because yeah, they're complex morphology, and I think well, we can do a lot of even stratigraphy with bryozoans, but we miss a lot of taxonomic work, and that hampers the use of bryozoans for these aims. But yes, Cucurcella is so curious and special that and since sections can be really very well recognized and well, I think we can find even more species with worldwide distribution, but this is just a question of taxonomy.
Mm-hmm. Yeah, I agree. It's one of the easiest ones to recognize.
I see there was a remark from Stephen. Andrej, very nice talk. Do you ever see stromatoporoids associated with bryozoans in your Ordovician material?
I have just one sample, which is interesting. I'm not sure whether it's bryozoan or stromatoporoid or tabulate. Well, yes, I think I have seen also some clear stromatoporoids in my material, but I have to check. Yeah.
Okay.
I have a lot of corals, tabulate corals, and Lugos corals as well. So it's, for Ordovician is also quite unusual.
Actually, I have a small question about your materials from Vasalema quarry. Actually, yeah. I remember Kluger, since he published a paper about reef and mount in this quarry, but I didn't realize that I didn't remember that he mentioned anything like just you show the bryolite. So I just have a continued question about this bryolite. Are they how big are they? The size? I mean, if they build reef, and then how big is this colonies? Yeah.
Do you mean the bryolites? So the largest one I have collected is about 20 centimeters in size.
Okay, so really small. That's why maybe it's very easy to overlooked by the colleagues.
Well, for bryozoans, it's really giant, because bryozoans usually are within 1 centimeter in size, or even less. So many smaller ones are about 1 millimeter in size. So 10 centimeters, 15 centimeters, a really large size for bryozoans.
Yeah, okay, thank you.
Yeah, okay, welcome.
There was a new remark from Stephen. He would very much like to see images of these things associated with the bryozoans. There may be a way to show correlations of paleogeography if they dispersed in the same way.
Okay.
Any more questions or comments? That does not seem to be the case. In that case, we go on to the last talk in this session, which is a keynote by Joe Budding on a new open marine Middle Ordovician Lagerstätten from Wales. Go ahead.
Okay, thank you. Just sharing the screen. Okay. All right. This is something completely new. It's actually discovered during lockdown. So it's because we've been stuck at home here in the middle of wonderful Wales, we've actually been forced to work on our local rocks. So sometimes there are surprises when that happens. Okay. For anyone who is playing bingo on which diagrams you'll see repeatedly, here was an extra point for you. But this is the classic Sepkoski curve and so on. But we've been studying, in many cases, the diversification, and in terms of the actual increase in diversity. But as Alicia pointed out yesterday, that's actually just a symptom of what is really going on. What we're really seeing in the Ordovician radiation, as I still prefer to call it, not an event, is a radical change in the structure of ecosystems and the way that diversity is structured throughout the marine realm. And so a symptom of that is the diversification. But we're doing this based largely on the shelly fossils, brachiopods, bryozoans, and so on, simply because we've kind of had to. But we know from the Cambrian faunas, particularly the Burgess Shale type faunas, that if you just look at the easily preservable components, you're only seeing a small part of the true diversity and a small fraction of the true ecosystem. And this is now sort of very standard knowledge in, when you're studying the Burgess Shale type faunas, that this entire richness that we see here in the top is mostly hidden. In that this, for the Burgess Shale, is a completely normal assemblage of trilobites, brachiopods, hyalids, and so on. And it's only when you get the exceptional preservation that you start to understand what the true diversity was like and how the ecosystems worked. So, for the Ordovician, we've been getting quite a large number of new Lagerstätten in the last 20 years or so. And these are starting to fill in a lot of the gaps, but there are problems. So in the Cambrian we have, I think it's, Bob Gaines counts over 50 Burgess Shale type faunas now, and most of the world. And they're all representing the same sort of environment and large parts of the ecosystem to various degrees. In the Ordovician, we have a wide range of deposits, which are representing a very wide diversity of environment types, paleoenvironments, and fossil assemblages. So it ranges from things like glacial lagoons and extreme deep water environments to single turbidity flows. Llandeckly rocks is extremely shallow water on the side of a volcano. And all of them are interesting. They all show facets of the ecology that we wouldn't have without them. But in terms of being a direct comparison with what was happening in the Cambrian, they're all very specific. They're all little windows into particular environments that don't actually correlate precisely with what we see from the Burgess Shale, etc. So when you take out everything that isn't directly comparable, which doesn't show how the continuity goes from the Cambrian to the Ordovician, we're really left with two deposits in the early Ordovician before the Gobi really got going. And of these, the Avangam is very hard work. There's some very nice stuff there, but it takes a lot of hammering to get much out of it. And the Fezwata, of course, has produced spectacular fossils, and many of them. But the ecology is strange, and we know it was nearly polar, and most of the assemblages are sort of very low diversity individually, and they add up to give a much more sort of complete picture, but it's odd ecology. So this talk is about trying to fill the rest of that gap a little bit for the Ordovician. And we're in right in the middle of Wales, for those who haven't been here. This little patch here is the Baltoscandia inlier. And it's about a 10 million year interval going from the upper part of the Darawilian, the Artostone through Murchisonite retiosculus, and into the base of the Sandian with the Gracilis bryozoan. And what we have in the built-in layer is this 10 million year sequence which records the entire evolution of a volcanic island complex. And it has fossils all the way through, and we have several sites with at least some degree of exceptional preservation. So Llandegli rocks is one that's always forgotten, but is a unique fauna from extremely shallow siliciclastic conglomeratic facets, mostly volcanic ash and so on, preserves spectacular sponge and echinoderm fauna, and lots of other things as well. But we're not seeing the truly soft things. We don't have the really soft-bodied animals there at all. Llanfawr is the opposite extreme. It's relatively deep water black mudstone, pyritized fossils. And this has, again, a lovely fauna. It's sponge dominated, but with other things as well. But again, it's something slightly aberrant. We basically don't see trilobites in these beds, which are absolutely normal for this area. So it's a little unique ecosystem that is being preserved. The Holothurian bed in the middle here is much more representative ecosystem, but only limited preservation. Again, we're not seeing the really soft-bodied things. We have echinoderm, sponges, paleoscalicid worms, but nothing truly soft. So today is going to be talking about this new site, Castle Bank, which fills in a lot of the gaps and is much more spectacular than the others. So this is right in the middle of the sequence, Murchisonite bryozoan in the Darawilian, and from the declining phase of the volcanic island, where it's basically eroding away, but there's still eruptions going on with ash beds. So the quarry doesn't look like much. We've been working here for about 5 years looking at sponges, and it's only this year we've discovered everything else, which is slightly embarrassing, but there you go. We have approximately 10-meter section, which is mostly absolutely typical graptolitic shale. So siltstone weathering to sort of pale shale. We've got tough beds here. There's lots of volcanics at the top. But then there's this 2-meter interval, which has exceptional preservation, by which I mean complete sponges and then a lot more. But within this 2-meter interval, there's a band of about 20 centimeters, which is really quite extraordinary. So this is a little log of it. Again, I said about 10 meters are actually exposed. And there are bentonites, ash beds, but this particular interval it's got a graptolitic shale background, sort of laminated mudstone, full of graptolites, but there are event beds in it as well, up to 2 or 3 centimeters thick. The event beds actually carry on further down too, throughout this 2 meters. But in this case, some of those event beds contain dense assemblages of soft-bodied Burgess shale type preservation. So, when I say soft-bodied faunas and exceptional preservation, this is the sort of stuff I mean. I'll go through the rest of the fauna individually, but it's fairly spectacular material. And throughout the talk, the scale bars are 1 millimeter, unless they're stated otherwise. So most of it's pretty small. But first of all, the normal fauna. And when I say normal, I mean about as normal as you can get. So at this age, graptolitic shale, it's full of Murchisoni. Didymograptus Murchisoni, I mean there's 4 on that surface. It's packed full of them, as you would expect. This time interval, when you get it, it tends to be abundant. It's not as abundant as this thing though, which is possibly the most boring brachiopod in existence. It's a lovely thing really, but you have to start blanking it out after the first 10,000, otherwise you go slightly mad. It's just, the surfaces are covered with this Hepatobolus macula. It gets up to 2 or 3 millimeters. And then one common trilobite, Aegitinus corndensis, which in the built-in layer occurs in all environments and is by far the most abundant in the range that it exists for. The first sign that there's something unusual is that almost every specimen we find of these is complete, not just a molt, but with the hyperstome intact and the 3 cheeks, and usually less than 5 millimeters long. The adults get to about 10 centimeters. So this is really quite unusual. We've got a sort of nursery situation where almost all of them are being buried alive. The rest of the sort of normal, if you like, shelly fauna is, again, absolutely typical. I won't go through the list, but you can see there's rare examples of bryozoans, a few different brachiopod species, Thenothalus, that sort of thing. but it's all completely normal in terms of the groups and the diversity. It's not high diversity, it's just what you would expect for this environment. Which is a good thing, because it means we're seeing what is probably an absolutely typical marine community for this time and this sort of environment. What is the environment? Well, the best clue, apart from the sedimentology, is probably the echinoderm fauna, which is very limited. All we've got are 3 specimens of this starfish, which gets up to about 3 millimeters wide. It's a new species, never seen it anywhere else. And possible fragments for a second one. But back in 2013, we worked with Bertrand on studying the ecological distribution of the various echinoderm groups through the entire built-in life sequence, and found that there's a really distinct difference between the shallow water faunas, which are dominated by the more derived modern type groups like really diverse crinoids, various starfish, echinoids, and so on, and the deep water faunas below storm wave base, where you have Stylothodon dominated fauna. And between the two, there's this interval where you get almost nothing apart from rare starfish, asteroids. And so, given the fact we have no Stylothodon plates at all and not a single crinoid ossicle, and based on the fact we have graptolitic siltstone with stalking with little event beds, and where it is in the sequence, we're pretty certain we are in this intervening depth, which is slightly too turbulent for the deep water faunas, but not turbulent enough for the shallow water ones. So somewhere around storm wave base, which is exactly the same sort of environment as the dominant Burgess shale type faunas in the Cambrian. So, don't worry, I won't concentrate on the sponges too much because I always talk about them. But I will say that they are spectacularly diverse. Like I say, we've been working here for about 5 years on and off collecting these. And really beautiful preservation down to the little the oscular spines, the marginalia around here. This thing is just 2 millimeters tall, but you can see the skeleton is actually really nicely preserved. They're all very delicate things, not few skeletons. These are all ones that have to be buried alive to be preserved intact. And we're estimating the diversity is more than 40 species, but to be honest, I have so many boxes of these downstairs that I haven't worked through. I honestly don't know. It's too many. But we're seeing mainly photomonaxenids, so sort of Cambrian type, typical of the Burgess shale type faunas, but also an increasing number of rarer but more diverse hexactinellid type things. So these really complex walled and interesting looking sponges. In contrast to the reticulosans, the very simple hexactin variant sponges that normally dominate this sort of offshore environment are very rare and hardly any demosponges. So it's an odd fauna, but very diverse. And going on swiftly onto the arthropods, because that's what everyone gets excited by. We're getting an increasingly diverse fauna, and it's very, very varied. So it includes various things that are typical of the Burgess shale type faunas, the isoxis-like carapaces, for example, and this sort of vaguely Ranchiocaris-like thing with the trunk coming out the end, but also some much more problematic things, lots of little bivalve carapace through the arthropods with their trunks preserved and bits of antennae and so on. This one we really do think is an opobiniid, and you'll notice Steve Pates is one of the authors, but we've just got the one specimen so far, so we're not going to be categorical until we can absolutely prove it, but it's got a lot of good features. That would be the first one outside the Burgess shale. This we think is a branchiopod crustacean. Again, there's more detail there than shown here, but it's got the pair of appendages at the front. And this, well, this one is rather interesting. It's 3.5 millimeters long, and there's a surprising amount of detail when you get into it. So you can see that the legs are preserved. We've got what appears to be a spinous mandible at the front here. There's an antenna, we think, coming across here. And we're not going to say much about this at all at the moment, except we really want another one, and it's got 6 legs. So yes, I'll move swiftly on from that one. But there are lots of other things as well. I mean, every time we go, we're finding more little crustaceans, arthropods of various sorts. And This one, for example, has a quite distinctive shaped carapace. You've got the trunk coming out the back, and at the front there's again spinosed appendage with well, something looks like a sort of slightly raptorial thing. But we have no sort of equivalent for this this type of preservation really in the late Ordovician, so it's all new. Okay, moving on to worms, because there's a lot more here than just arthropods. Paleoscolecids, of course, they are the group that you would expect to turn up in the conservate life strata or offshore ones at this time, and we do get them mostly. Well, we have a few semi-articulated things with quite nice preservation. This is the posterior termination here. We haven't got anything complete with proboscis yet, but sure, it's only a matter of time. And we have numerous cuticle fragments. I mean, I've got a box full of things like this, which we haven't got into electron microscope yet because we're stuck in our house in the middle of Wales and nowhere near a university. But we do have probably 5 or 6 species at least which you can separate based on the sort of the microscopic characters. So lots of paleoscolecid diversity. And also lots of other worm diversity. So priapulids, which are a very common group in the Burgess shell-type faunas in the Cambrian, are also quite abundant here. We normally get a couple of specimens a day when we go out to sample the best beds. And vaguely ortoya-like things with expanded posterior parts, proboscis area here, another proboscis, and several of them preserve the gut as well, the dark line down the center. We also have other worms, so polychaetes are much less abundant in these faunas generally, and also here. But this one doesn't look too promising to start with because it's sort of a bit mangled and it's degrading at the both ends. But you follow this end round and we have a scolecodont, and the other end where it is rotated flat, we've got bunches of bristles coming off the side. So it's something with chaetae and scolecodonts, it's a polychaete. We have other things that we're fairly certain are polychaetes as well, but waiting for detailed studies. So this one is 1.5 millimeters long, but it has these rather nice chaetal bundles coming off the on this side and coming off here as well. This one, much smaller chaetal bundles. There is a diversity of forms and things that we think at this stage are polychaetes. Okay, what else? Well, until recently, chinorhynchs had no fossil record at all. Then they turned up in the early Cambrian and a couple of places in China. The Xinjiang biota especially apparently has a diversity of them. But we seem to have them here as well. So again, a few different morphologies. This one, well, I'll just say this one verse is extremely small, just sort of fraction of 1 millimeter long, but it's got the sort of plated structure. It's got the sort of the hairs or spines you can just about make out, but we need a bigger microscope. This one, well, when we were working on this locally, someone popped up in a nearby village saying, "I've got an electron microscope in my bungalow, would it help?" So we've actually put this one into an electron microscope, and we have quite a lot of detail coming out of this. And all the features that we can see support that this is actually a chinorhynch. It's got organic spines on this sort of plated-like body. We've got terminal spines coming off the end. We've got a different structure over here with the broader spines around the collar region. And the proboscis area, this part of the flank, is very strongly wrinkled, suggesting it was much softer than the rest of it. So again, this is very early days, but we're fairly confident that these genuinely are chinorhynchs. And there are hundreds of other little worms. I mean, there are some surfaces where you get the right layer, you can get 4 or 5 little wormy things on the surface at once. And many of them, well, some of them might be other things entirely, but many of them probably are worms. We have no idea what group a lot of them are yet. There's just too much. Which is a nice problem to have, but yes, there's a lot more work to do, and there's a high diversity. Soft-bodied mollusks, yeah, we've got a few things like that as well. So we have possibly the first Ordovician Wiwaxiid sclerite. We're hoping that some of these will fall out of acid dissolution at the moment, but it looks good from what we can see with this one. And this rather cute little thing here. We're not entirely sure what it is, but it seems to have little eye spots on the end of stalks. And the closest we can get is something like a nudibranch sea slug, which I believe has no fossil record at all, apart from the ones that have shells. So again, we've only got one of these so far. It's early days, but we would love to find some more of that, just because they're so cute. But we have a range of other stuff as well. So tentaculate filter feeding things. Again, note the millimeter scale bars. We have a long sort of stolon-like structure, a little conical body, and then these very delicate tentacles coming off. And if you zoom in on the tentacles, you can see that they have very fine sort of cilia-like structures. They're actually a bit too big for cilia, but still sort of 50 micron long, little structures coming off them. It's ridiculous preservation, some of this. And this is carbonaceous as far as we can see. We haven't got elemental mapping yet, but it's got to be carbon. It's just compression fossils of tiny, tiny little soft-bodied elements. Hemichordates, yep, we seem to have probably the second definite record of zooids in a graptolite, and possibly the third as well now. This is a dendroid graptolite with the silvery branches you can just about see on the rock, quite faintly preserved. And this colony has 5 or 6 little zooids inside the branches with what appear to be, we're going to be working on this in detail, but we're pretty certain there are traces of the lophophore here and the plug at the front, cephalic shield. Cephalic shield, thank you. It's handy having a graptolite expert next door. And we also have lots and lots of tubes of a thing that Kozlowski called Byronia, based on something that was from the Burgess Shale, but in the Ordovician it may be something different. And we have soft tissue in some of these, including, every time we see it, a pair of tentacles and what may be a cephalic shield here as well. So possibly these are also hemichordates, but again, it's something we're working on. There we go. Possibly cnidarians. Apart from things like Sthenothalus, the only likely candidate is a couple of specimens of this thing here, which has some very odd structures going on. But those structures are very reminiscent of what you get with the mesenteries and the musculature of sea anemones in Xinjiang. So we've never seen anything like this elsewhere. We want some like they have in Xinjiang, where the tentacles are all sort of splayed outwards radially so we can actually see what it is, but possibly we've got soft-bodied cnidarians, I'm not sure yet. And likewise on ctenophores, there's a whole range of problematic things have been recently suggested to be all part of a continuum with basal ctenophores, including these sort of sessile forms to start with. We've just got one specimen of this turned up about a week ago, and it doesn't look like much. It's a strange sort of creature, a few millimeters tall, but when you zoom into here, there are fine transverse bars going all the way across most of these petal-like structures. So it's very similar to things like Cyphus octum from Burgess Shale, this tulip animal. And if this is a ctenophore, whatever it is, interesting, but if it is a ctenophore, then it probably fits in somewhere around here in this sort of ancestry of them. So there's an awful lot of things turning up. Vertebrates, the only thing we've got definitely so far is a conodont bedding plane assemblage. No soft tissues yet, but the conodonts themselves are quite rare, so I suspect we'll get more of that at some point in future. But quite nice preservation. Occasionally you see an isolated conodont on a bedding plane, but normally you just don't find them at all. But one day we might get more. And of course, like in all of these conservat lagostatin, there are things that we don't know what to do with yet, things that are really tantalizing. It's like this one really does look like a radiodoct, a non-lacaridid. And there are features that really do suggest it might be, but we're not going out on the limb and saying it is until we get a second specimen and confirm some of the fine details in these appendages and so on. Some of these other ones we're having great fun trying to interpret, but again, we need more information to get very far. But there is a lot of problematic material and things we just don't know what to do with yet. And there's a whole other avenue of that in that this morning I got the first photos were sent through from Sweden. Elise Vallet and Seb Willman have been working on SCF dissolutions. And in their words, it is very promising. So we have lots of bits of graptolite, or terraprank at least. But note the scale bar here, this is about 150 microns wide. So it's either one of the very slender things like Maslagographtus, or it's something that we haven't seen yet in the fauna. But really nice preservation. Chitinozoans probably, I guess that is as well. But then there are bits of cuticle with all sorts of interesting ornament on it. So, like I say, this is the first that I've seen of it, and that was about an hour before we started this morning. So there's probably, hopefully, a lot more to come from the SCFs. And I'm trying to give you some vague idea what the sort of distribution of the diversity is. It's extremely sort of back of the envelope sort of thing, just trying to work out what we think we've got so far. But the fauna has a very large chunk of sponges in the diversity, but also an increasingly large chunk of arthropods. And that's probably going up faster than the sponge diversity is. Hemichordates are a major group and much more diverse than we were expecting, and then lots and lots of other things as well. So there is, as far as we can see, the full range of diversity that you might get in the best Burgess shale type faunas. So for conclusions at this stage, it's not a concluding stage to be honest, because this has been done by two of us working intermittently when the sun is out in lockdown for a few months. And it's 3 miles away from our house, so it takes a little bit of effort to go and do it. But we've barely started. The amount of rock that we've gone through is quite small. We've been working out how you actually find these fossils. And little details like when you first split it, they're invisible. And it takes 30 seconds for them to oxidize and become dark so you can see the things. That took us a little while to work out. So we've barely started, and there is a huge amount more to do. But we can say that this is the first conservat Lagostata from the middle Lower Ordovician that is directly comparable to the Cambrian Burgess shale type faunas. We've got the same sort of environment, we've got the same range of taxa and the same preservation styles. And in fact, the sedimentology with this sort of sponge-dominated background beds and then little event beds with these soft-bodied things in is so strikingly similar to Chengjiang. It's just on a slightly smaller scale. The fauna is extremely diverse, and both in terms of the species, we think we've got 140 or so far, and who knows where it's going to stop. But also in terms of the major groups, the phyla and so on. And we're getting soft tissues in groups where they're very, very rarely seen. And in terms of what we can say about what it's telling us, one striking thing is that a lot of the fossils are tiny. And we don't think this is a bias because you do get bigger fossils as well in the same beds. But most of the worms, the chinorhynchs and so on, and the little arthropods are very, very small in the same way as they are in the modern, but which they're not in the Cambrian. So in the Xinjiang biota, there are the chinorhynchs are several centimeters. And here they're up to 1 millimeter or 2, which is what they are in the modern. So we may be seeing possibly a transition from the Cambrian type faunas into the more modern faunas by the middle of the Ordovician in terms of these soft body things. So I'll just want to just thank a few people. The site owner, Ben Duell, and his family are incredibly supportive. Ray the farmer for digging things up for us. And I should say that Lucy and I work mainly as independents here in the middle of Wales, so we've actually crowdfunded a very nice microscope, which if anyone's got the window sort of open, you can see sort of there. And we've got another one coming as well with camera lucida. So thank you to everyone who supported that. And also a little virus which forced us to actually work on these local rocks that we've been sitting on for 5 years and not noticing we had a Burgess shale type fauna. So that comes as a relief, and I'll stop it there. Thank you.
Thank you. There was already a question on the chat. Keith is asking you, any trace fossils on the event beds that could support the paleobathymetry?
None at all at the moment. Apart from we have coprolites, pellets of various sorts. I don't think we have seen a single burrow. which is odd. But then if it's a normal Burgess shale type sort of situation where they're being transported into a very relatively anoxic area or very low oxygen area, then possibly at the site of burial, there wasn't enough oxygen for in-fauna, but is carrying in-fauna in from a slightly different place. But no, we haven't seen any burrows yet, but we don't have polished sections, so there may be some sort of fabrics there later.
Okay. Ollie has made a chat here. Great material, Joe. The event beds are tophitic turbidites question. Creating an abruption Lagerstätten. Will you do SEM on the conodont assemblages? Look like you have beautiful Baltoscandia elements.
Thanks, Ollie. They're not tophitic, actually. They are just, they're micaceous, but they're basically siltstone. They're slightly coarser than the background, and it's probably just slumps from the side of the volcano, but they're not actually ash beds. So you can, the ash beds are much paler. It is kind of a brucium in the same way that all of the Burgess shale type faunas are, but we're getting the carbon film preservation and some pyritization and so on as well. So it's the typical sort of range of preservation. We would love to get someone working on the conodont assemblages, and actually we're thinking of emailing Ollie at some point, weren't we? Yeah. So yes, if you're interested, you'd be very welcome to help out.
Okay. There was another chat, which is, I should forget, I can see. Then it's Christian, fascinating talk regarding the possible upapinit. What features would you say might support that identity?
That's something we are still working on, but there are several details that you can't see in the sort of overall image. So that the trunk, the nozzle type thing at the front is very finely annulated in the way it should be. We have the mouth and the anterior digestive tract apparently preserved in the right orientation and position. And there's one or two other things that Steve Pates is looking at as well that he thinks are quite distinctive features. So we haven't got the tip of the nozzle, which would have the spiky bit on, or the tail fan at the end, but there are other features there that do seem to be fairly convincing. But we'd love to have a second one, and we probably will need one before we go public with that. But yes, we're fairly sure that it's at least likely. Yeah.
Any other questions or remarks?
I have a question about the taphonomic and the preservation of this fauna, because as I know, since you are working with all these Lagostella, spanning raising from the lower Ordovician to the late Ordovician, and as you know, I'm working on the carbonate stuff, and we noticed that during the Ordovician we have really a revolution in the shallow marine. We see the disappear of like a flat pepper conglomerate. We have a lot of like hard ground and the deepening of cementations, which means we really have a totally different environment in the revolution in the sea floor. But I just wondering if you can see any change in this in shell, because you are working on similar I mean lithology from late Ordovician to from early Ordovician to late Ordovician. For example, in Anji formation, are they show any difference between the fauna you just mentioned in terms of taphonomy and preservations?
It's a huge question, because people often sort of conflate mudstones or siltstones all being similar, but when you start looking at the ecology in the Ordovician, they are so different. Every site that we go to has slightly different ecology, at least. In many cases, very different ecology. So the Anji biota is black mudstone also, but it's much deeper water. And the fauna is completely different to what we see here, apart from some of the sponges are quite similar. But even in the built-in layer, from 1 meter to the next, you can have very different taphonomy, very different ecology. It's surprising how much variability there is. So we don't see anything that I would call a sort of systematic change, because there are so many variations from the early Ordovician onwards. There's no sort of general type that you can expect to stay the same. Does that make sense?
So it's basically the environmental difference variations.
Yeah, there's huge amount of variation within these offshore mudstones and siltstones. Just slight differences in the grain size, in the oxygenation level perhaps, but also just inherent in the ecology. We see things that look like identical sediments, you know, a few hundred meters or half a mile apart, and they are very different ecologies. And there's no obvious reason for it. We think it's just part of the Ordovician radiation that you start getting segregation of ecosystems on a much smaller scale than we saw in the Cambrian. And if this was in the Cambrian, we could predict half of the groups that would turn up just by comparison with all of the other sites. But in the middle of the Ordovician, we have no idea what's going to be found next.
Yeah.
Any more questions or comments?
Birger has raised his hand.
What, sorry?
I think Birger has a question.
You are, yeah?
Yes, I raised my hand at least. So two questions. We talked yesterday about possible events. We have this expansion or radiation, and then if there are any major events on the way. And if you read older literature, it looks as if there was an event at the Arenig-Schlangenwirn boundary. Can you relate where your Lagerstätte approximately is in the stratigraphy relative to that old concept?
This is in the middle Fanvern. So
okay.
The old boundary was the base of the Artistone, wasn't it? Yeah. Right. So it's about a biozone and a half below.
Okay, so it's not that, it's later. And one other quick question. Have you compared with the Winnicek Lagerstätte? Can you say any differences or similarities or?
Yeah, Winnicek's totally different. And basically that is an impact crater, as you know well. It's shallow water. You have this sort of peculiar environment where lots of arthropods were going into to reproduce, but it's not an open community. It's in the same way that Sum is a very isolated environment, so was Winnicek. So it's extremely impressive Lagerstätte, but it's telling us about that one little corner. But there's virtually no similarity. And the only thing I think we've got which is shared are little serraticarid crustaceans. But that's the one thing they've got complete. And we just have fragments because they're living in the surface water 50 or 60 meters up and molting up there. So we get very nice fragments, but we don't get the complete ones of those.
Okay,
so
no, very nice work. Thank you.
Thank you.
I think we are out of time. Now it's a coffee break. So I have been baking apple pies in my kitchen here. Danish apple pie. Anyhow, we will reconvene at 10:30 Danish time, and I think the official time is 2 hours earlier than that. So there's a short break here. Thank you everybody for keeping on time. It went very smoothly.